I request the participants to kindly be seated. Distinguished dignitaries, esteemed guests, invited speakers, delegates, members of faculty, students, print and electronic media. A very good morning to you all. On behalf of the Laboratory Animal Scientists Association, LASA, India, and GITAM School of the inaugural session of LASA-Con 2025, the 13th International Conference of LASA, being hosted at the lush, green and academically vibrant campus of GITAM University. LASA-Con 2025 is being held on the theme Preclinical Insights from Animal Studies on Drug Discovery and Translational Research. This conference is envisioned as a vibrant forum that brings together LASA members, eminent researchers, academicians, and professionals engaged in preclinical and translational research. It aims to facilitate the exchange of innovative methodologies, highlight recent scientific advancements, and foster critical discussions that meaningfully contribute to drug discovery and development. Today we are privileged to be joined by eminent speakers from across the globe who will share their invaluable expertise in preclinical research. Ladies and gentlemen, on this momentous occasion, let me take the pleasure of inviting the distinguished dignitaries onto the dais. With great delight, I invite the organizing secretaries of LASA-Con 2025, Dr. G. Suhasan, Associate Professor, GITAM School of Pharmacy, Visakhapatnam, and Dr. M. Gerald Mahesh Kumar, Chief Scientist, CSIR, CCMB, Hyderabad, to grace the dais. I am glad to invite the conveners of LASA-Con 2025, Dr. Nitin Yadav, Associate Professor, GITAM School of Pharmacy. I am glad to invite the Chair of Visakhapatnam to grace the dais. I deem it a privilege to invite Dr. R.K. Shaktidevan, Secretary. It is my honor to invite Dr. Vijay Singh, President, Laboratory Animal Scientists Association, to kindly grace the dais. I am immensely pleased to invite Gitanjali Padmanabhan, Honorable Pro Vice Chancellor, GITAM Institute of Medical Sciences and Research. It is my privilege to invite an erudite administrator, Professor Y. Gautama Rao, Honorable. I deem it a great privilege to invite a distinguished scientist renowned for his pioneering Honorable Chancellor, GITAM University. A visionary leader in science and entrepreneurship and the founder of Shanta Biotechniques. He led the development of India's first indigenous privilege to invite Padma Bhushan Dr. K.I. Varaprasad Reddy, the chief guest of today's program, to kindly grace the dais. To rise for the invocation with Vande Mataram. Vande Mataram. kindly join.
हरिवरासनं स्वामी विश्वमोहनं हरिदरीश्वरम् आराध्यपादुकं हरिविमर्दनं स्वामी नित्यनर्तनं हरिहरात्मजं स्वामी देवमाश्रये शरणमय्यप्पा स्वामी शरणमय्यप्पा शरणमय्यप्पा स्वामी शरणमय्यप्पा शरणमय्यप्पा स्वामी शरणमय्यप्पा शरणमय्यप्पा स्वामी शरणमय्यप्पा शरणकीर्तनं स्वामी भक्तमानसं भरणलोलुपं स्वामी अरुणभासुरं स्वामी भूतनायकम् हरिहरात्मजं स्वामी देवमाश्रये शरणमय्यप्पा स्वामी शरणमय्यप्पा प्रणयसत्यकं स्वामी प्राणनायकं प्रणतकल्पकं स्वामी सुप्रभाञ्चितं प्रणवमन्दिरं स्वामी कीर्तनप्रियं हरिहरात्मजं स्वामी देवमाश्रये शरणमय्यप्पा स्वामी शरणमय्यप्पा शरणमय्यप्पा स्वामी शरणमय्यप्पा शरणमय्यप्पा स्वामी शरणमय्यप्पा Okay, thank you.
I thank Dr. Gattadi Srikanth, Assistant Professor, GITAM School of Pharmacy, Hyderabad, for the soulful rendition. It is 2025 to deliver the welcome address.
Respected dignitaries on the dais, Honorable Padma Shri Professor V.S. Chauhan, Chancellor, GITAM Deemed to be University, Padma Bhushan, Dr. K.I. Varaprasad Reddy Garu, Founder, Santa Biotechnics Limited, Honorable Pro Vice Chancellor, Geetajali Padmanabhan Madam, Honorable Pro Vice Chancellor, Professor Y. Gautam Rao, Dr. Vijay Singh, President LASA, Dr. R.K. Satyadevan, Secretary LASA, Dr. Gerald Mahesh, Vice President LASA, Dr. Harishankar, Dr. G. Suhasin, Dr. Nitin Yadav, the conveners of today's program. Of the dais, Professor Amrita Ahluwalia, Dean, Queen Mary's University of London, Dr. Monty Triplalak International, Dr. Penny Hawkins, Head Animal Science Department, RSPCA, Professor Urvi Chatterjee, Professor University of Calcutta, Professor S. Ganpati, Ex-Rector, Andhra University, and our former Dean, GITAM School of Pharmacy, and other distinguished members. It is my privilege and honor to invite you all to this mega event, LASACon 2025. That is 13th International Conference of Laboratory Animal Scientists Association, India, which is being organized by GITAM School of Pharmacy and with association with LASA. The theme of the conference is Preclinical Insights from Animal Studies on drug discovery and translational research. The event is being organized for the first time by a pharmacy college. GITAM School of Pharmacy, being a NIRF 33rd ranked institute among the pharmacy schools in India, accredited by NAC, NBA, feeling very proud to host this event. The conference is witnessing the multidisciplinary scientists and researchers belonging to various fields like medical, pharmacy, veterinary sciences, and other life sciences. The speakers are representing from various international and national universities, institutes, industries, and research labs. The deliberations of the conference will help the young researchers, professionals in preclinical research and drug development. I extend my heartfelt thanks to LASA for giving us this opportunity to organize the event. I also thank GITAM management for rendering continuous support to GITAM School of Pharmacy for organizing this event. With these remarks, I once again welcome the August gathering to this event. Thank you very much.
Thank you, sir. LASACon 2025 to present the opening remarks.
GITAM University, Padma Shri, Professor Virendra Singh Chauhan, Pro Vice Chancellor, Professor Y. Gautam Rao, Pro Vice Chancellor, Jimsar, Professor Gitanjali Padmanabhan, Dr. Vijay Singh, LASA President, Dr. Jaral Mahesh, Vice President, LASA, Dr. Shakti Devan, Secretary of LASA, and all other distinguished guests of the dais, eminent speakers, delegates, researchers, academicians, industry professionals, and dear students. On behalf of GITAM School of Pharmacy and LASA, it's my privilege to welcome you all to this LASACon 2025, the 13th International Conference of LASA India. We are delighted to host this prestigious international conference, bringing together the experts and researchers from diverse backgrounds to exchange their knowledge, idea, and the recent advancements in the laboratory animal science and preclinical research. We are pleased to welcome you all to Visakhapatnam, fondly known as the City of Destiny. We hope you enjoy the richness of this conference and the natural beauty and the warmth of the Visakhapatnam. The theme of the LASACon 2025 is the preclinical insights from the animal studies on drug discovery and translational research, which actually gives the role of ethically conducted and scientifically robust animal studies in the drug discovery pipeline and in the translation of the basic research into meaningful clinical applications. Here, the scientific program includes the plenary lectures, keynote address, invited talks, and the oral and poster presentations covering the emerging trends, ethical guidelines, animal welfare practices, and technological advancements in the laboratory animal science. We are confident that these sessions will stimulate the meaningful scientific discussions and foster the future coordination and collaborations among these individuals. I take this opportunity to express my sincere gratitude to the GITAM University for its constant encouragement, institutional support, and the excellent infrastructure which made this possible to organize this conference successfully. I also grateful to this LASA India to extend their extending their support throughout this conference. I thank all the delegates for their enthusiastic participation. I encourage each one of you to actively engage in these sessions, interact with the experts and fellow participants, and make this most learning, networking collaborations provided by this LASACon 2025. Once again, I extend my warm welcome to all of you and wish this LASACon 2025 a great success. Thank you.
Prestigious conference. May I now request Professor Y. Gautam Rao, Pro Vice Chancellor, GITAM, to kindly address the audience.
My colleagues from the School of Pharmacy, School of Science, from both Visakhapatnam and Hyderabad campuses, industry experts, industry leaders, researchers, students, should I say all protocols observed. It's my privilege and pleasure to invite you all to this workshop, the 13th International Workshop of Laboratory Animal Scientists Association. I would like to thank the association for choosing GITAM as the venue for this year's conference. I have been watching my colleagues for the last almost a month plus. They have been working so hard to ensure that this conference becomes a success. So I would like to thank the team led by Professor Geetanjali ma'am, Srinivas Garu, Raja Garu, and the other faculty members of the School of Pharmacy. GITAM believes in doing cutting-edge research. Naturally, research has been on the top of the agenda for the last 6 years or so. In line with that, the university has been spending crores of rupees on its infrastructure, also recruiting faculty who have gone through the portals of premier institutes and papers. So I would request the researchers and faculty who have come from other universities and other colleges, please do find some time and go around and see what are the facilities GITAM has, and if you feel you would like to utilize them, probably you can contact our principal of the School of Pharmacy. Now, coming to the importance of pharmacy in the Indian context, I think everybody is aware India is called the pharmacy hub for the entire world. And I'm sure the memory is still very strong with us. Not long ago we had that COVID pandemic, and you have seen how the Indian scientists and the Indian pharma companies worked day and night to come up with co-vaccine and Covishield to ensure that not only the Indians are protected from COVID, but also millions of people across the world. So we enjoy that kind of respect as the pharmacy hub of the entire world. Of course, of late, due to some political decisions of other countries, the pharma industry, like a few other industries, has been going through turbulent times, and I feel it is only a temporary phase. Once we overcome that, we will have another great time in terms of our exports to both developed and developing countries. In fact, I would like to make a special appeal to the researchers, the students of pharmacy. You are very lucky to have a galaxy of stars amidst of us. Please take full advantage of this. Do raise questions, do seek clarifications, develop networks with these galaxy of stars, and I'm sure you are going to gain significantly, and that will be memorable something in your career so early. With this, I would like to thank the organizers once again for giving me this opportunity. I wish you all the best. I wish you wonderful deliberations, and I'm sure the people end of the day tomorrow. God bless you all. Thank you.
Thank you, sir, for your inspiration and encouragement. I now invite Dr. R.K. Shaktideva.
Very good morning to everyone. On behalf of Laboratory Animal Scientists Association, I welcome you all for this wonderful platform, providing the opportunities, joint hands with the Laboratory Animal Scientists Association, so to conduct this event in this part of this country. So we are very happy. We are looking forward to interact with all the scientists, researchers, industry experts, collaborators, all the eminent professors here. So with that, I wish all the best. I request all the members to participate all the sessions and get more insights from the international conference. Thank you everyone.
Vijay Singh, President LASA, to address the audience.
25. This is the 13th conference we are continuously hosting across India, in every part of India. Visakhapatnam, you might have discovered, is very beautiful city with a lot of good beaches and lot of good places to see. And it is so beautiful that all of us, those who have coming, especially from Delhi, have become nocturnal. So we are active in the day and also we are active in the night. And here is the proof if you don't believe me. So I went at 5 o'clock to the beach side, and these are the random people we recognized from the Lhasa Kon. There were many others whom I did not recognize, and we were all doing fun, including yoga and Sirsasana and Mayurasan, having fun. So thank you very much for the leadership of Visakhapatnam GITAM University, Pro-Chancellor Sir, and also Chancellor Sir V.S. Chauhan Sir, for helping to clean our brain and clean our lung. So both things we are cleaning here, and this is the another proof. We are joined by our another colleague from veterinary faculty, beautiful dog. He also got interested down the line, so we cannot ignore him, and he also joined. This Lhasa conference and Lhasa have a very good history. It started in 2004 by our founder, Dr. Ramachandra Sir, who is sitting in the front row, and our Secretary General, Dr. Kadri, who could not come here. They had a vision, and because of that vision, we are here. It is my request to all of you, please greet them with a round of applause for starting such an organization. At the time, we were only 11 founder members, but now we are closing 1100, nearly touching 1100. And it is so good to see that out of 1100, nearly 730 are present here. And this is giving me immense pleasure that our laboratory animal science is getting a traction, not in India, but everywhere. It is so proud to see the Padam Shri and Padam Bhushan on the Lhasa Kon dais. Chauhan sir, thank you very much for giving the time. Padam Shri and Padam Bhushan is really great for the Lhasa Kon dais, and we are so proud, so honored and super happy that sir you gave us the time. And without respecting our senior, we cannot progress. So it is a good idea. We always go with our senior colleague with the advice. We also request to have a very productive conference. Those who are senior, please don't shout on the juniors. They may be having some mistakes, some problem, but it is good idea to guide them rather than having the hard shoulder. That is not acceptable. Lhasa is a friendly society. We do science, but we do science with the fun. And when you do science with the fun, it goes into your head in a permanent memory, not the temporary catch-a memory. So permanent memory, if you want to make, you have to have fun, and fun mixed with the science. And that's what we are looking for. For any society to progress scientifically, we must have a mouthpiece or we must have a journal. We have a Journal of Laboratory Animal Science, and this Journal of Laboratory Animal Science doing now very, very good. It is being indexed into ISSN, NAS, UGKL, and Copernicus. And great recognition of this goes to Dr. Nagarajan. I don't know whether he is here or not. Nagarajan Perumal, he is from, where is Dr. Nagaraj? He is not here. He is very shy. If he is here, he will not stand. Yeah. But he is doing great job as editor-in-chief, and meticulously we are publishing continuously, and we are even getting the, especially thank my secretary Shakti to continuously help in reviewing, Rahul Thorat, who is also not here, and my executive committee, so that we do all scientific rigorous our review before we get it published. Also, I request in future or now, we will give importance to our journal. For any award, any consideration, our first preference, as our Prime Minister says, make in India. So, we will give consideration for junior. If you have any data, please do publish with us. If you have any problem, we have editorial assistant, and we also have people who help us in writing and putting the manuscript together. So I request all to also publish and cite, because until unless we start citing the paper, we will not have indexing into Scopus. So this is another challenge we have, that before we go to the PubMed indexing, we start citing. And it is taking, our journal is taking very good. Next is to facilitate young people. We are instituting many awards. We had only Lifetime Award, but we have started with Creativity Award. We have started with Lifetime Achievement Award, so that youngsters who are doing excellent can be recognized. By now, you know, the foreign people are putting a lot of trouble to us. USDA, Department of Agriculture, is putting so many complications that we are impossible for us to import the animal. So now it is a time that we should stand on our feet rather than looking to the West, have our own model, have our own preserved things, so that we can have our own. We are trying very hard with the DHD and USDA, because they have unilaterally challenged their sanitary and phytosanitary certificate, which is creating trouble for us. So for this particularly, we are invigorating and stimulating our junior people with the awards, different category, which we will announce today, and also later we are also thinking, so that you start thinking, not looking to the West, but make in India, our Prime Minister vision to make Viksit Bharat by 2047. And also, as I told you, we should not forget our leader. Last time we had a great successful conference in Mumbai, and I especially request Dr. Arvind Ingle to stand up, and a round of applause. It was a great successful congress, and he is also the past president. Not only the successful congress, we always have a guest, this editor journal every time. We will have this time, and we will have Nitin Yadav, our colleague, who will be doing this journal with a full publication. And last time also we had a great number of publications and great feast of science in Mumbai last time. Also, we are having a lot of a skill development program. So LASA India is supporting 6 regional workshops, and I am so happy and proud that we are not limiting to India. Our skills are being asked. Recently, Padam Shri sir, Chauhan sir, we have been asked by Vietnam government to continuously handhold us to the society journal as well as the training program. Indonesia, Malaysia, Vietnam, as well as Kenya and Ghana. We have gone there, host the training program, and they are asking us to continuously host the program across India. We balanced that 6 workshops, this which is supported by LASA, had every corner, and especially we give support to those who have never been supported by LASA, so that every corner and nook will have the benefit of laboratory animal science. And this is thanks to our senior for thinking about this skill development program. I request all of you to make this LASA success a movement. Success a movement in the way of publication, success of the movement in the way of participation, success of the movement in way of encouraging the juniors to publish, to speak, and also to travel and learn and come back and do the great science together back in India. And with these words, I thank you all, and I wish to thank my LASA Executive Committee. Whatever I said is not my words, it is the word of LASA Executive Committee, and also this is the word of LASA family. LASA is a family. When we get next hosting program, we started to prepare like we are preparing for a next Bharat. Bharat, you understand in Hindi? Preparing for like a marriage function. That we will have good time for next 2-3 days together, meeting people, networking, dancing, and having fun. Say this motto with me: good science with good care. Please little loud. Good science with good care. Okay, with these words, thank you very much for the opportunity by the GITAM leadership, Swasini Madam,
Nitin sir, Harisankar sir, and my beloved friend Dr. Mahesh. Friends, we should not forget how much he is contributing. And special thanks to Padam Sri and Padam Bhushan sir for gracing the occasion of Lhasa conference. Not only that, 730 present 1,000 mice, rat, guinea pig and rabbit. So please accept from us, not from the all 730 delegates, but the mouse which are housed in our facility. Good science with good care. Jai Hind, Jai Bharat.
Thank you, sir, for the encouraging message. We are truly privileged to have with us.
After Vijay's, I think before I say anything, first of all, welcome to Visakhapatnam, welcome to GITAM. I'm not going to say anything about GITAM University. I think all of you here, you've heard, you had a glimpse of it. Please, before you leave, go and have a good look at what the GITAM administration has been able to do in the last 6 years. You have to see yourself and make your own assessment. But after Vijay's forceful welcome and talk, I'll simply say Vijay's place is not Lhasa. If I may say that, Vijay, you belong to Parliament. And you belong to Parliament, you represent all of us, and you represent all lab governments. Anyway, again, I welcome all of you. I didn't realize when I was asked in Hyderabad to come to Lhasa meeting, of course I gladly agreed, but I didn't realize this is such a large family as I see myself. I'm very happy to see a very large number of you, and I hope also 2 slides that Vijay showed, I was thinking maybe this meeting or this talk I'm giving, or I will give later on, should be done on a beach, not in this room. So anyway, enjoy yourself. Those of you who've never visited Visakhapatnam, please have a good look at the city. Extend your stay by a day or 2. You will love this city and come back. Many of you are young. You will look for future career. You won't find a better place than GITAM to come and start your career. Thank you very much. Okay.
But for the benefit of non-Telugu people, I switch between both English and Telugu. Kindly pardon me. I am not a scientist. I do not belong to this kind of seminar or symposium. I am the odd man out here, but still I have 2-3 reasons to come here to stand before you. Fundamentally because that by providence I came into healthcare sector, though I am trained as an engineer in electronics. I came into life sciences healthcare in 1993. Until then, I was an R&D scientist in electronics engineering. I was in defense R&D, and after that I switched to industries promotion. But because I was in the science healthcare sector for last 35 years, I have little right to come and stand before you. Second reason is I was awarded doctorate here by your chairman, erstwhile chairman, MMVS Murthy Garu. I have gratitude. As part of my gratitude, I accepted the invitation. Though I am not well versed with your science, I have peripheral knowledge. I do not know much, but I can speak about the importance of this science, laboratory animal science. I have gone through that, though I do not totally understand intricate matters of lab sciences, lab animal sciences. I am affected one. So I have another reason to take up this invitation to express what I have gone through. This is a foreign science to me. I have jotted down some points. I will read it for you. And where I feel what my pain, it is not written, I will speak to you. So once again I go into my written message. Good morning to everyone. Distinguished guests, esteemed scientists, faculty members, delegates, and dear students. Though my background is not from pharma or life sciences, I am here to stand before you with my hands-on experience in biopharma sector. Incidentally, you may be knowing, until 1993 there was no biopharma industry in the country. First biopharma company in the country is Shanta Biotechs. Dear students, you are the future of pharma and medical research. It is my great privilege and honor to address the gathering of experts in pharma, biotech, and lab animal sciences. Here, stalwart Dr. Chauhan is here. In the initial stages of Shanta Biotech, I had several discussions with him, advices from him. He was very helpful to me. And my gratitude to you, sir. As the founder of Shanta Biotech is a company dedicated to harnessing science for public health, I have witnessed in close quarters the extraordinary journey of drug and vaccine development in India. This journey, which brings life-saving products to millions, is intrinsically linked to the discipline of laboratory animal sciences that you are celebrating today. My experience, I told you that when we were developing recombinant DNA-based hepatitis B vaccine, and the second product was interferon alpha 2B. You all know that we have to go through several stages of testing. ICGB, I think they were guiding the protocols at that time. Institutional Biosafety Committee is the first gatekeeper for whatever development we do. After that, GEAC, Genetic Engineering and some other abbreviation, I don't remember correctly, GEAC. After that, preclinical studies, that is animal studies. So all these things were not known to me, but my scientist group, they were dealing with that. While they were dealing with the science, I had to deal with the social problems. At the time, when we were going through the animal studies at National Institute of Nutrition Laboratory in Hyderabad, there was a society called Society for Preventing Cruelty Against Animals. They were kind to animals, good, I salute them, but they should be more kind to humanity. Suddenly they came and evacuated the animals that we are going through. with the premise that we are troubling the animals, we are cruel to animals. They thought that they should liberate them from the test laboratory. They left them in Srisailam forest. Next day they died. Is it kindness or foolishness? Those animals were not trained to move around. For a long time they were kept in cages. They were taken very good care. Timely meals, timely taking the blood sample and then giving them some nutrition, whatever it is. They were maintaining them. Suddenly when they evacuated them and left them in the forest, they don't know how to move also. They have become a sumptuous dinner for the tigers in Srisailam forest. So our entire episode of making all these studies gone waste. Our 9 months of hard work have gone waste. Because we approached court, we won the case, we argued with the so-called kindness society towards animals. That is mandatory by science. It is not my wish that I have to experiment on animals. In the protocol written, we have testing on Swiss albinomites and some primates. The primates were liberated by the Society of Cruelty Against Animals. Maneka Gandhi was in charge at that time, and I approached her. The first dialogue was, "You scientific bastards." I bore the brunt. I said, "Madam, your language is very filthy. This is necessity. We have to go through this animal testing. Without that, we can't ensure safety and efficacy. Unless they are proven, I cannot give it to humans. You have gone through vaccination, your child also gone through vaccination. How they have come? Only after testing. She doesn't want to live another. You scientific bastards," again she repeats. "You are very cruel to animals." Then what should I do? I ask her. The solution is not stopping the experiments. Give 100 crores to the society, then you will be let free. So 100 crores will solve the kindness. That is what we call foolishness of our leaders. Anyway, we have gone through that. But kindly remember, you are the first line of defense for any drug or vaccine that comes into use. You are in such a vital stage, point to observe whether this drug is going to have safety, whether it is going to give acquired protection, then only it will pass on to the next stage. The development and deployment of vaccines and drugs are fundamentally relies on rigorous preclinical testing. The complex pathways of drug metabolism, toxicity, and therapeutic benefit must first be mapped in relevant animal models. This meticulous work is the central focal point that transitions a compound from the research bench to human clinical trials, ensuring patient safety. Therefore, the laboratory animals are very essential not only for the efficacy, potency, and safety testing of drugs or vaccines that are manufactured. The laboratory animals should be of high quality and well maintained for the accurate results. The regulators will rely upon the clinical data and thereafter and teaches the child in adult is safe, effective, and free from hidden risks. Still will be average 60 to 70 kgs. We give LD 40 in acquaintance with the latest science. In those days, we were giving LD 40 times dose of what human beings are given as lethal dose. If that survives, now after the society has taken, okay, they have done some good work on the safety of animals, you should be knowing. If they found an alternative method of, instead of bothering it with LD 40, some other mechanism to prove that lethal dose level, at what level it is dangerous to human beings. Every quality control test for potency and toxicity relies on established ethical and robust animal models. Long before clinical trials, it is in our labs that we get the confirmation of immune response. We look for the right level of antibodies or cellular immunity that will be able to fight phase. During the R&D phase also, animal models are instrumental in moving a concept from a test tube to a human trial. From our lab only, optimal and safest dose are determined. We should not view normally the feeling or belief is animal sciences is a supportive mechanism. In my opinion, that is the vital and fundamental first stage of ensuring the safety and efficacy, and it should not be treated as a supportive mechanism. Laboratory animal science is not a support function, see it as a first line of defense for public health. Innovator, the next generation of life sciences depends on data integrity and workflow precision. The primary bottlenecks in research are no longer conceptual, they are operational. Across academic and industrial labs, promising work increasingly stalls, not because of hypothesis, lab skills, technical expertise, and professional competencies. Build a healthy Bharat and be proud that you are in the healthcare sector. And now I switch to my mother tongue for the benefit of my own Telugu students who are here. ఆరోగ్యమే మహాభాగ్యం, అంతకన్నా మించిన భాగ్యం ఇంకోటి లేదు. ఎవరు ఏమి సాధించినా ఆరోగ్యం లేకపోతే వాళ్ళు ఏమి సాధించినట్టే లెక్క. ఆ ఆరోగ్యానికి మనం ప్రకృతికి దగ్గరగా ఉంటే ఆరోగ్యం బాగానే ఉంటుంది. మనకు తెలియకుండానే ప్రకృతికి దూరం అయ్యాం. ప్రకృతికి దూరం అయితే పర్లేదు, ప్రకృతికి ద్రోహం చేస్తున్నాం, దాన్ని బాగా హింస పెడుతున్నాం. దాని పర్యవసానమే మనకు వచ్చిన రోగాలు. అండ్ పర్సన్ ఈజ్ నాట్ ఎట్ ఈజ్, హీ విల్ గెట్ ఇన్ టు డిసీజ్. మనిషి ప్రశాంతంగా ఎందుకు లేడు అంటే ఉన్నదాంతో తృప్తి లేదు, ఏదో సాధించాలని, ఏదో పరుగులు తీయాలని. మనిషి ఆల్వేస్ ఈజ్ గోయింగ్ త్రూ స్ట్రెస్ఫుల్ లైఫ్. ఈ కారణంగా ఈజ్ నాట్ ఎట్ ఈజ్, సో ఈజ్ న్యాచురలీ ఎంటరింగ్ ఇన్ టు డిసీజ్. అయితే దీనికి ప్రత్యామ్నాయం ఏమిటంటే మందులు. ఆరోగ్యం ఉండడానికి మందులు, ఆరోగ్యం పోగొట్టుకున్నప్పుడు మందులు, ఏ రోగం రాకుండా ఉండడానికి మందులు, జీవితం అంతా మందులు. ఇవాళ మనం తినే ఆహారం కన్నా వేసుకునే మందులే ఎక్కువ. ఆహారం తగ్గిపోయింది, మందులు పెరిగిపోయాయి. వీటికన్నిటికీ కారణం ఏంటంటే మనిషి తను ఉండవలసిన స్థితిలో ఉండకుండా ఎక్కడెక్కడికో ప్రయాణం చేస్తున్నాం. పక్కింటి వాళ్ళని చూడంగానే మనం మార్స్కి వెళ్ళి చూడాలి, చందమామ మీద నీరుందో లేదో చూడాలి, మన పక్కన మన కుళాయిలో నీళ్ళు లేదు, మన ఇంట్లో నీళ్ళు రావట్లేదు, దీనికి ఏ నదులు అనుసంధానం చేస్తే మనకు జీవితం చక్రంగా గడుస్తుంది అన్నటువంటి ఆలోచన తగ్గిపోయి మార్స్లో నీళ్లున్నాయా లేదా, చందమామలో నీళ్లున్నాయా లేదా అనే పరిస్థితి వచ్చాము. ఈ విపర్యాల వల్లనే మనిషి అనేక స్ట్రెస్ఫుల్ లైఫ్లో ఉన్నాడు. కాబట్టి అనేక రోగాలు వస్తాయి. రోగాలు తప్పవు. మనిషి ప్రోగ్రెస్ అవుతున్నాడంటే ఆర్థికమైనటువంటి ప్రోగ్రెస్ అనుకునే స్థితిలో పడ్డాం. కాబట్టి ఎన్ని ఇండస్ట్రీస్ చూస్తేనో, ఎన్ని జాబ్స్ చూస్తేనో, ఎంత సంపాదిస్తేనో అదే జీవితం అనుకునే రోజుల్లో మందులు తప్పనిసరి, మందులు ఇన్ఎవిటబుల్. మీరు మంచి హెల్త్ కేర్ సెక్టర్లో ఉన్నారు, అది చాలా అవసరం. ఆ సెక్టర్లో ఉన్నందుకు మీరు గర్వపడండి. దానికి తగ్గ నిజమైన నిజాయితీ, ఇంటిగ్రిటీతో ఆ సైన్స్ని పాటించి ప్రజలు కాపాడండి. నమస్కారం.
Thank you, sir. I now invite Dr. N. Harishankar, sir, to say a few words.
Very good morning again. This is Lhasa Lifetime HC Balachandran. I request Dr. C. Balachandran come onto the stage. I request Dr. Kailavan to accompany him. 2025. Dr. C. Balachandran, born on 28 July 1957 at Managuri, Tamil Nadu, is an eminent veterinary pathologist whose distinguished career spans over 4 decades of exemplary service in teaching, research, administration, and in institution building. He earned his BVSc in 1980, MAs in 1983, released from Madras Veterinary College and Tamil Nadu Agricultural University. And he has PhD in 1993 in veterinary pathology at Madras Research in avian pathology. He was awarded the University Gold Medal for the best PhD thesis. Dr. C. Balachandran joined Canvas as assistant professor in 1980, became associate professor in 1988 and professor in 1996 and retired on 31st July 2000. He also served the university with distinction as Vice Chancellor and Registrar. His visionary leadership was instrumental in the establishment of 5 veterinary colleges and research institutions across the world. A charter member of research. He played a pivotal role in developing laboratory animal science education in India, notably facilitating the first philosophy category certificate course conducted in oncology, nanomedicine, veterinary therapeutics. His work on traditional Siddha and Ayurvedic formulations, nanomedicine-based interventions, clinical evaluation of animal populations, and experimental cancer models significantly advanced the preclinical toxicology and therapeutic research in India. Dr. C. Balachandran's outstanding contributions have been recognized with more than 100 plus prestigious honors, including FAO Fellowship in Avian Pathology, Fellowships of IAVP, NABS, and the ASAW, the Tamil Nadu State Science and Technology Award, IAVP, Dr. Nemichand Jain Lifetime Achievement Award, several national oration and thematic lectures, and the Dr. C.M. Singh Centenary Oration Award 2022. In recognition of his exceptional achievements, unwavering commitment to education, research and service, and his transformation in India, he is pleased to confer upon Dr. C. Balachandran the Lifetime Achievement Award, honoring a career that has profoundly enriched the veterinary science and biomedical research in India.
మన టీచర్లను మనం గౌరవించుకుంటే అది మన సాంప్రదాయాన్ని గుర్తు చేస్తుంది. We should honor our teachers. Majority of the veterinarians are here who were trained under the able leadership guidance of Dr. C. Balachandran. Now, he will hand our Shakti Devan will hand out the citation to Balachandran sir. And Padma Bhushan Varaprasad Reddy sir will hand over the memento to Balachandran sir.
President Lasa, Dr. Vijay, Singh, Saktidevan, Gerald. So as time passes by, we realize the importance of careful use of the animals and how we can reduce such kind of things. Then in the CPC and IAC committees, they have regulated this use of animals. And in toxicopathology, what we pathology study is something different from the toxicology pathology we could understand in later days. It is regulatory compliance. So involved, and we have to comply with the different countries' requirement. And I also underwent training on toxicology pathology and Dr. K.S. Rao, an eminent toxicologist. In academic research, it is something different because it is proof of concept. So we design differently, unlike that of the regular CROs what we are doing. Because here the student have to do the work individually, whereas it is supported by many personnel in case of here. So in this, I would like to say a few words about the genesis of FELASA and international training. That is, this federations of European Laboratory Animal Science. Sciences that is offering this training. So we have formulated with the LASA came with this proposal, and then they has approached as Tamil Nadu Veterinary Animal Sciences University, the first university in the country, Now about 14 such universities are there in India. So then it was well supported by fellows on the NASA side at the time. Dr. Ramachandra, Dr. Syed Padri, Dr. Sachdevan, and also fortunately we also had from the National Institute of Animal Welfare, Dr. Ashwini Kumar. So he also a tripartite agreement was signed in Hyderabad. Our Vice Chancellor, Dr. R. Prabhakaran, I was a minister at the time. So he agreed to be in the discussion and agreed. And also we started this program from 2013 onwards. And I have served as a nodal officer for this for 4 years. I really thank Dr. Prabhakaran for his magnanimity to making me as a thing. So then, so far we have conducted about 13 such programs. 316 people have been on ICLAM. So then it was recognized later. Now I hear that there are 21 deployments are there in ICLAM and 5 founders and 8 de facto members. I am happy that it is progressing well. Being the Vice Chancellor, then I extended from the laboratory animal for mice, rats and guinea pigs. We have extended to large animals also and rabbit giving separate facilities at our Madaram. There is in another livestock farm. So that has been working till date. And we have during that period we could establish BSL 3 containment facility in which we have the IVC cages for rat lice and also separate facility for chicken. This is open for the people to use it at a reasonable cost. We can approach that. So that way, why we have gone into this program, international program, this accredited, it can go anywhere it is recognized. With that view only we have done this thing. And quite once again thank the NASA organizers for giving the opportunity and really I appreciate after hearing the inspirational talk by Dr. Vijay Singh. Kudos to, they are not simply with the conducting conferences, they are going beyond training to national now into international and then recognizing young people who are contributing on the research side. That way it is being extended and I am also happy to be making India. So we should have our own, that is why we established this facility that should be excellent. So we thank the organization. Thank you, sir.
Thank you very much for the nice words, sir. NASA Fellow Award 2025. The first innovative award goes to Dr. Mahesh Ahare, please.
Round of applause for Mahesh Ahare.
One more award has been initiated about the new Creativity Award. That goes to Dr. Shivaraman.
Unfortunately, Dr. Sivaraman could not able to join because of the flight cancellation, but he will be handing over formally. So this year, NASA Fellow Award, Dr. S.T. Muthukumar, and the Creativity Award for Dr. Mahesh Ahare. So both of them on the screen right now.
So, dear friends, ladies and gentlemen, the award is the predecessors or the speakers like Padma Bhushan publications. Like that, our Muthukumar and Mahesh are also publishing several publications in national and international journals. So, this is the right time to honor them by giving a standing ovation, please. I request all of you to please stand and give a round of applause to this Kavango. Thank you very much. Now I hand over the mic to the Professor Madam.
I request our principal sir to felicitate Padma Shri Professor V.S. Chauhan, Honorable Chancellor GITAM. I request the chairman to felicitate Professor Y. Gautama Rao, Honorable Pro Vice Chancellor, GITAM deemed to be University. I request the chairman to felicitate Professor Gitanjali Padmanabhan, Honorable Pro Vice Chancellor, GIMSA. I request the chairman to felicitate Dr. Vijay Singh, President Plaza. I request the chairman to felicitate Dr. R.K. Shaktidevan, Secretary LASA.
Scientists committed for welfare of laboratory animals. Laboratory animals helped us in understanding the underlying putative mechanism behind the development of diseases. Now, it is our duty to serve the laboratory animals by keeping them with proper care and management. This pledge that I will remain committed towards welfare of laboratory animals and devote time for this. I will devote maximum working hours voluntarily for welfare of laboratory animals in the facility. I will develop alternatives to the lab animals and minimize their usage in the scientific research. I will initiate the use of alternatives to the lab animals with myself, my colleagues, my animal facility, and my research institutions. I believe that the institutions of India use lab animals, encourage the use of alternatives to lab animals and welfare of them. With this firm belief, I will propagate the message of use of alternatives to lab animals and their welfare. I will encourage, make them devote their time for welfare of laboratory animals. I am confident that every step I take towards welfare of the lab animals will help in minimizing their usage in scientific research.
Respected dignitaries, guests, colleagues and participants, a very warm welcome to GITAM Deemed to be University. We extend our deep gratitude to the GITAM management for their constant support, our President Shri Bharat Garu, Vice President M. Gangadhar Rao Sir, Chancellor Sir Professor Chauhan, Vice Chancellor Professor D'Souza, then Geetanjali Madam. These are all here. Then I also want to express my heartfelt appreciation to the Principal, School of Pharmacy, who is the Chairman of this LASACON 2025, Professor L. Srinivas, for his vision, leadership and continued support in making this event actual possible. We are grateful to the executive committee members of LASA, all the committee members, because we were in touch and we were doing so many discussions during this period since long. So special thanks to Dr. Vijay, the President of LASA, Dr. Shakti Devan, Secretary, and then Vice President Dr. Gerald Mahesh, and finally Dr. Hari Shankar sir. He was continuously, these people were behind us, so obviously we finally are here and then we are going to have this event. I convey my warm thanks to all the invited speakers for accepting our invitation to share their expertise and provide hands-on experience. Yesterday we had hands-on training also, so I want to just thank all of them. Today this special thing is there that two of our highest civilian awardees, Padma awardees, one is Professor Chauhan and then one is Vara Prasad Reddy sir. These people are there. I'm very happy. One of them is my guru, so that's why I can't even ask for more than this. So it's more than enough for me, because now my confidence level is from here to here. So I thank both of them. Then finally, my deep thank to my colleagues, including faculty members and non-teaching staff who are working day and night. Yesterday night I was on phone, some students were at airport, 11 o'clock, 12 o'clock. So my deep thank to all of them, volunteering teams and everybody, because without these people, I always say, this also Professor Chauhan only told me, without student, we can't have university. These people, you are the backbone. I always say, all the functions. Without you people, we can't stand. So that's what I want to convey. So after that, we also acknowledge the excellent support provided by GITAM hospitality team, finance team, security and maintenance teams. Because without these people, this planning, execution, nothing is possible. Nothing. So that's what these people were always there, always. Then last, not the least, and closest to our heart, the student participants. most important part of all universities, all conferences, even workshops also. So a great thank you for all the students, whoever are here, our volunteers, our college students. So to our school, School of Pharmacy students, I give.
जनगणमन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तब शुभ आशीष मांगे गाहे तब जय गाथा जन गण मंगल दायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
and visited the facility in Hyderabad. So those of you who are growing up now probably won't realize how difficult or how brave or how far thinking some of these people must have been. Today India can take pride that it makes more than 50% of all vaccines of the world. But that's the time when India was just beginning to start. So those of you interested, and many of you should be interested, and I say that with clarity because you might think you're doing pharmacy or you are interested in lab animals protection, but there are no boundaries to science anymore. So biomedical sciences, pharmaceutical sciences, pharmacy sciences, life sciences are seamless one into another. So don't think if you're doing a course that you can't do. This is what people like Varaprasad, Krishna, and many people. And as I said, India probably is not probably, India definitely is the largest producers of world-class vaccine at very affordable cost. I am a chemist by training. When we started our career at ICGB, it wasn't very clear which way ICGB will go, but very soon I think we're mandated to look at infectious diseases, and I think we continue to focus on that. And malaria was one of the first disease after the dengue and hepatitis. and tuberculosis later on when I became the director. So I'm actually not even very keen to show you slides because that consumes time. But still to make some sense of what we did, and some of us were very younger than what I am today, but we didn't have that much idea. In fact, the malaria field itself vis-a-vis vaccine grew up only in late 80s. So when if I start talking about this in detail, I think it will be very confusing. But just say to you, those of you know or do not know malaria cycle, when the infection begins, when a female mosquito which is laden with a parasite takes a blood meal from a human who not infected at that time, and while sucking the blood out, that is the time when the parasite enters the blood, in the human body. And first 7-8 days, depending on the species, there are many species of malaria. Depending on which species there is a silent malaria infection which is growing inside the body. It's impossible to detect malaria at that time, although you are infected. And that is what the first stage. So those of you are curious about any disease or anything that you see around, the question you will ask, the malaria parasite inside the blood first enters only the liver cell. So here is the scientific question: what is so special about liver cell that the parasite would enter? And many questions. And some of these things were simply not very well studied at that time. And for example, even in India today, to study the liver stages, maybe 1 or 2 labs is very difficult. After from the liver stage is come into the blood, you see the red circle there. At the blood stage, when the parasite has changed from what it was in the liver, this is the time when you can detect malaria, this is the time when you have fever and so on. So I'm not going to bother you with the details. But when the blood stage parasite comes out, then some population of the parasite goes into sexual differentiation. So far the cycle is asexual, and then the parasite, which is called gametocyte, comes out, and once again when a female mosquito takes the bite, the parasite will enter. And this is how the life cycle goes. So you can imagine it's not easy to be parasite also. It has to go to many changes. So, but if you look at the cycle in something like malaria, which is very different from a viral vaccine, you could interfere at the liver stage, you could interfere at the blood stage, you could interfere at the last stage, and so on. So all these vaccines have been tried. In fact, there is a vaccine which is in the final stages in the phase 3 trial in Africa, developed at Oxford and produced in India. And I'll come back to that if I get time. So these vaccines, as I said, in early 90s, there were no vaccines, there were only experiments. So we entered this field, and looking back today, I can say we entered without knowing what we were entering into. And the idea of that we will look at a malaria vaccine sounded very good, but we didn't realize the challenge, and I just articulate some of the challenges to you. But first of all, next slide. Yeah, so this is what you see on the left, the yellow, is the parasite, expanded picture of parasite. We know much more of this than we knew in 1990s. After the genomes were sequenced, genome of human, genome of parasite, genome of mosquito, all are available to you at a click of a mouse. So you can do as much work as you want. Some of the organelles that you see, the blue, now we know at the time of entry, on the right you see the parasite trying to enter a red blood cell. It first attaches, then reorients, then gets inside, then hugely multiply within 24 hours. From one you can have 5,000 parasites. There's much activity going on inside the red blood cell. You would think if you could stop the interaction of the parasite, first interaction of the parasite with the red blood cell, you probably will stop malaria, at least at the blood stage. Similarly, if you stop the first parasite entering the liver, stop malaria. And these are the rationale for trying to make vaccine. But if it is so then why don't we have a vaccine? Why didn't we have a vaccine? That's the complication. So first of all, there are more than, now we know there are more than 5,000 genes that malaria parasite expresses. So 5,000 is huge number. And this is not a lecture on malaria, it's just very complicated. If I go to the next slide. So this information was beginning to come, and what you see at the top is a cartoon of a major also wants to live, and human also needs to find a balance. So most of these proteins are very polymorphic. So from one strain to other, even in the same cycle inside a human, you will find many proteins, many strains of the same product, many versions of the same protein. So how do you make a vaccine? What you need, something highly stable. But if you see the end of the top protein, the red end is what we found out, many others found out, is the business and business in ICDP environment. We define these things. And the next thing would be, naively we thought if you make these proteins, and that's where the strength of ICDP of that time, that we thought we could make any recombinant protein. Not knowing what lies ahead. So these portions of the next slide. And we got international attention because a student like you were, through your hard work, we were able to make very high quality protein. I'm not going to go into detail. This is the time when we got money from Bill Gates Foundation, we got money from European malaria vaccines. So we attracted a lot of international money on primarily the ability to make very good protein. These are E. coli expression. And I have a message to give in. So we characterize these proteins. This is a key slide for the message. Now the basic research production of a protein in the lab, identification, characterization, did not take that much effort. Of course it was not straightforward. And they're disulfide linked, they have to properly fold, etc., but we were able to do that. And now, in the vaccine, now we can't shove protein just like that. Lab animals, of course, are indispensable. And I think Varaprasad Reddy rightly said, he said, I hope some of you caught that word. He said, good quality experiments. So you can do lab experiments in lab animal, but you can do the that, as I said, Shanta Biotech was the first one to produce, and many other things. These are beginnings of many things. So when we went to the ICMR for permission to get a protein, in fact, in the ICMR, people did not know how to handle the recombinant protein that has not been introduced in humans before. So we had to produce the point and show it to the ICMR. So anyway, we did all that. So this took us about 20 years. But fortunately, we were funding. And what happened, what we had to do then, produce a lab process, because these proteins were not made by anybody else before in India, or for that matter, whoever was doing vaccine work, that we had to develop a process. Now, we didn't have a process developing lab at ICD, even with the money. So we constructed a lab, we reconstructed our animal house. Finally, we were able to produce a 10-liter scale of protein production. Today, if you were to do it, and I have written this to DBT and to ICMR, today if you were to do it, the best way to do it would be, if this is a serious matter, whether it's a tuberculosis vaccine or dengue vaccine, and industries are now much more advanced in India. They are mature. Without industry, try not to do translational. I would even say that without industry involvement in the beginning, even the funding agency must not consider. These are the lessons that we've learned. Because the industry which will then help you to do GMP production, and all of you know this, if you do preclinical good studies, there is no escape finally to translate if you did not have facility to make GMP grade product, no matter whether you're developing a drug or you're working on a vaccine. So that cannot be avoided, and that's what we learned. Fortunately, a company in Hyderabad,
very well-known company, Bharat Biotech, which was also starting at that. We were all starting at the same time. Produced the vaccine, and to cut a long story short, we also, those of you, and I hope many of you would get into vaccine development, because there are many vaccines yet to be made. The vaccines alone, protein alone do not work. You need a carrier and what we call adjuvant, something you want to make a formulation in. And if the academia simply does not have access to proprietary adjuvant that Matrix M or many others, they are the ones which will go forward. You will be very lucky if aluminum hydroxide will work for you. So many vaccines have aluminum hydroxide, but we will be very lucky for a protein-based, a subunit-based vaccine to go forward with hydrogen. So if you have industry on board, most industry in India and elsewhere have matured. They have their own adjuvant system. Formulation itself, you think in a sitting lab I can formulate, but you really can't. Formulation, looking at animal itself is a long process. We learned harder way. But anyway, finally we were able to produce the vaccine at Bharat Biotech, managed to get money, and Singh in Bangalore, a company. And this is a long process. I'm just squeezing it into a very small chapter. We finally, what guided us was the animal experiment. We could show in animal that both the proteins put together produce robust antibody responses. And here comes the preclinical development versus going forward. What we saw in animals, in humans that was not reproduced. So the antibody response we saw in animals were simply not high enough in humans, and suddenly the whole project, as it were, not collapses, but it just comes down. Now you worry, either we just leave it, say it's bad luck, we spend so much money, what we saw in animal didn't happen in humans, we can't do anything. Or you look at the science at that given time. In my lab, we are now working on malaria furiously, basic science. Another protein called MSP3. We did a lot of work on MSP3. MSP3 was taken forward by French group as a vaccine candidate. And what was missing in our construct, MSP3 had that. In other words, so those of you who are all scientists, so you can get B-cell responses to get antibody responses. But you need T helper cells, epitopes to make the antibodies. And unfortunately, the red portion that you saw in the first part didn't have any T-cell epitopes, which we realized later. And then in the lab, we fused MSP3 portion with the portion that I showed you earlier and produced what we call a fusion protein. This is about 10 years back. Yeah, about 10 years back, 15 years back. And when we looked at this fusion protein that produces antibody both for the MSP3 as well as for MSP19, and we thought it was a very good candidate. We went back to Department of Biotechnology and we again got funding. So again the large amount of funding, and we were then in this case we learned some lessons about dealing with the industry. We went to Zydus Cadila. Zydus Cadila produced the MSP3 protein, and we were then given money to do the human trial. Now, before this we had to do a lot of, I can't say there, but at least were 30 slides to show that what you've done is better than before. Otherwise you won't be able to do the trial. Unfortunately, by the time we were ready with 10,000 vials of formulated MSP3, the COVID showed up. And the moment COVID showed up, a whole lot of people stopped working and so on. So I'll just cut the long story short. The final stages of 10,000 vials are still in the deep freezer. And now we are trying to revive this. So we are very confident. And some international group also have shown interest, but only after we show the safety in humans. So the safety, animal safety is animal safety is important. Good quality animal experiments are important, but of course the crucial for any drug vaccine is the safety in humans. And you can't you cannot, no matter how much AI develops, no matter what happens, without the good quality animal experiments, I don't think we'll produce things which were to humans. So the society like Lhasa, gatherings like this, should remember that hugely important to have good quality care animal houses. They are expensive to run. If you run them well, they are expensive to run. But I think that's the core fair. So that's where I think we are. I obviously don't want to show you the point here is this, that the Indian industry, Indian biotech industry, and particularly Indian pharmaceutical industry, is as mature as anywhere you will get in the world. I dare say they are even better. So most vaccines that we see, which are in the experimental stages, are being produced in India. For example, Oxford has a very large group in all vaccines. They work on malaria, tuberculosis, dengue, you name the vaccine, Oxford group works on it. They do have that small GMP facility within the university. And if you were to plan a university which wants to work on vaccine, that's the way to plan. At the first stage of vaccine, you can produce in large amount. Malaria even have a human challenge model. India still has not brought the human challenge model. Another time when I speak with you, then I'll tell you advantages of having human model. For example, today, if we want to take this vaccine any further, we cannot sidestep the human challenge model. And it's not very difficult to bring human challenge model. So in many ways we are very good. In many ways in India we are still deliberating. Can it be done? Of course it can be done. And therefore most vaccine that you'll see, even TB vaccine or dengue vaccine, which is now in human trial, all being experimental vaccines are being made in India. So that I don't want to name the companies, there are many, including Shanta, which are absolutely pioneer. And so therefore the younger people sitting here, the message to you, you just need to step out from what you think you are being trained for. Be brave. Me as a chemist, I never had handled E. coli in my life. And we now realize that if we produce protein in E. coli, it probably won't go forward. And we'll have to express this protein in yeast. Because there's so much work now already going to be VLP, virus-like particle produced in yeast. But after this is where the GITAM, universities like GITAM and others should think of. If we have to work in these areas, that the incoming of mRNA vaccine, which changed the way we look at vaccination. I don't think there's any disease now which is worth looking at, and people are not already looking at mRNA vaccine. The future of vaccine seems to be mRNA vaccine. I don't think protein vaccine will be replaced so easily, but the future of exploratory research will be mRNA vaccine. But the key would be how to stabilize RNA. So there's enough so that RNA is stable. But I can tell you, we follow the field that there's mRNA malaria vaccine, mRNA tuberculosis vaccine, mRNA HIV vaccine, mRNA dengue vaccine. So the field is just exploding. I'm not saying that future will be only mRNA vaccine, but investigations and probably some cancer vaccine for that matter, the preventive, curative vaccine. So the field is fantastic to get into. This is the right time to get into, but one has to think of futuristic. But finally, I'll end my talk by saying that there are no boundaries in science anymore. If you are in pharmacy at GITAM or for that matter in pharmacy department, that's the best place to start vaccine. If you're life sciences, and most of these universities have these departments together. Many diseases worth looking at which the West is not interested. In fact, the West has gone the other way. They probably are more hesitant to have any kind of vaccine, let alone highly workable vaccine. So I would stop. There's no place better than India to start the research. Regulatory authorities need to figure out their own ways, but the industrial support in India is unmatchable. It's certainly not available at the cost and the quality and the cost in Europe or in Australia or in America. This is the best place to do vaccine work. Thank you very much.
I think this was the story. It's not easy. We can talk and then we can listen. It's easy to look at. Now, right now, the time, what he said, he showed HPLC. It's very easy to do these days. But earlier, just look back for 20 years. Nothing. Even still, so many universities are struggling to have HPLC in their lab. So that's what you can just imagine what was there 20 years or 25 years ago. So this work, all vaccine work, when nobody was talking about vaccine. Right now we all know vaccine just because of the COVID. Because we all saw, now everybody knows about vaccine, what vaccine is. Otherwise nobody knew. Malaria, nobody knew. So thank you so much, sir, Professor B.S. Chauhan, to be here. Because as a group, I have seen him working very hard, doing so much of hard work for not only for the universities, funding agencies, everywhere. So I learned so many things like discipline. So that's why just now he told that no need of lecture because we are already beyond the time. So he told no need, but still we have to listen to him because the story actually I feel, multiple times he usually says whether we have to go ahead, all their life. So that's why I don't want to leave these candidates. Maybe, I don't know I will be able to do that or not, but we'll take these things forward. So thank you so much. Thank you so much, sir.
Enriching and successful LASACon 2025. We will be breaking for tea and I request all to reassemble in 10 minutes. I request all the participants to reassemble in 10 minutes.
Professor Srinivas and Professor Harishankar for their very, very kind and wonderful hospitality. You all have been so very nice. I've had a wonderful visit here. I very much enjoyed seeing your beautiful city and very much enjoyed the interactions with the GITAM faculty and also the faculty at the medical school. So thank you very much for your time. So today I'm going to talk about sex as an experimental variable and discuss whether there's been any progress within the biomedical research environment. We know that as an issue it has been having growing profile. There's a great deal of interest in trying to correct a very terrible wrong that exists within the biomedical research environment. But before I speak about some of the work, I just want to highlight my disclosures. I have a number of disclosures here, but the one that is the most relevant for this presentation is that I was Editor-in-Chief of the British Journal of Pharmacology for the period 2016 to 2022 inclusive. It's relevant because I'm going to share with you some of the work that I did in that role with an international editorial board to raise the profile as sex as an experimental variable within the pharmacology community. But before I do that, I just want to highlight that I'm not just interested in addressing the deficit in consideration of sex in the experimental realm in terms of generating guidelines, but actually I'm very interested in sex differences that exist in health and in disease, but also in cardiovascular disease, pathology and pathogenic processes. And as a reflection, I've just listed here some publications that we've made in the preclinical arena, looking at mechanisms of regulating vascular tone and therefore blood pressure, processes of regulating immune function, acute immune responses and consequently atherosclerosis. I'm particularly proud of the work that we've done demonstrating that males and females actually use very different processes in controlling vascular tone from within the endothelium. They have completely different pathways And unless we study those mechanisms, it means that we're not necessarily treating women with high blood pressure with medicines that can work for them. But I'm also really proud of the work that we've done taking our basic discovery observations into the clinical setting. So we've done a number of studies looking at blood pressure in healthy volunteers and hypertensive patients, but actually we've also set up experimental medicine models of inflammation and the impact of inflammation on cardiovascular function in the lab. And I think one of the critical things for translation is making sure that you have this connection, as we heard from Professor Chan earlier, between your preclinical work and the clinical translational element. And I think if you work closely with clinical colleagues, it is actually possible for the discovery scientists to do at least some early clinical work. We did some really interesting studies where we looked at acute inflammation in healthy volunteers. And originally I went with the view from our mouse studies that women would show a lesser magnitude response to an acute inflammatory stimulus than men. And actually that was completely wrong. It wasn't direct translation from the mouse to man, as we heard often happens. But actually what we discovered is that of resolution might underlie some of the coronary artery disease epidemic that's now occurring in women, that they have some deficiencies in their pathways that most women actually have that protects them from coronary disease. And we've established responses. But today I'm here to talk about how we developed at the British Journal of Pharmacology policy documents to improve consideration of sex as an experimental variable in the preclinical arena, particularly with a focus on pharmacology. So I like to show this slide. It's a slide that I made when we were celebrating the 75th anniversary of the British Journal of Pharmacology for two reasons. One, because I think it was gratifying for me to have my name added to it. You see the very first editor-in-chief of BJP, Sir John Gadam, an absolute giant in the establishment of the early principles for pharmacologists. And you can see here the names of a whole host of gentlemen who actually look very similar to Sir John Gadam. And so you can see that and I was very gratified that in the UK that they decided to make a change. So embracing diversity in its fullest. So this actually are some metrics that come from the journal. They're a little bit dated now. As I said, I finished being edited in 1994 onwards up until 20. You can see the impact of the journal just stayed pretty steady for a lot of its history, slightly improved. It started improving even more in the during the editorship of my predecessor, Ian McGrath, who was very focused on identifying approaches for the Pharmacology International Editorial Board. We generated a whole host of policy documents, guidelines, to help our research community design best practice experiments that improved reproducibility and enhanced transparency. And this rising impact, it continued to rise actually up until 2022, I think. This green line actually shows the ranking of the journal, and there's a little dip here, but you can see it largely maintains its position. And I think this is a really, really important point that I'm about to make. So if you look here, in 95, there were 157 journals in the pharmacy and pharmacology category. In 2020, this had, you can begin to see what a massive task that is. So society-owned journals, they have a mandate to promote best practice, to disseminate best practice, but there are so many from another, and many journals do not have minimum standards for publication in them. And so the job of a general appreciation that there has to be dissemination of these guidelines from not just within one journal but across journals, and that is a very tricky task. So what's the imperative for some of the key issues? But I like to show this statistic because I think it's a really, it gives clarity about the rationale. So women are not particularly in the period between 1997 and 2000. 8 of them were withdrawn because they posed greater health risks for women, women with the treatment of common disease than men. And the reasons for that is that we do not understand disease mechanisms in women very well. And that is because they have been insufficiently studied. It's very, very complex. There are lots of areas where further research is needed. Liver metabolism is different between the sexes. Volume of distribution is different between the sexes. Fat distribution is different, and therefore for lipophilic drugs there is a big issue there. There are so many differences that it's absolutely imperative that we focus on doing basic research and making comparisons of different journals that they analyse across endocrinology through to neuroscience and pharmacology.